Thank you very much for the kind introduction and the invitation to speak today. Actually, I'm from the University of Galway. I'm a neuropharmacologist. The project started during the time when I was working at the University of Auckland, and we just finished the project recently. I'm going to talk about the drug that we think it's a really promising treatment for MS, but also maybe for other neurodegenerative disorders. As Almudena explained already, in MS, the role of the inflammasome, it's really well known and well studied. So this was a really good disease model to choose to examine the effect of this drug. The drug is undergoing clinical trials at the moment for eye disorders. But this is the first study what I'm going to present to you today, where we are examining the effect of the drug in a neurological disorder. So we don't need further introduction to the inflammasome. And as I mentioned, it's well established that it's activated in MS and also in the animal model, the EAE model of MS. The drug that I'm going to talk about, it doesn't target the inflammasome itself. It works upstream of this, and the way how it does it, it blocks the connexin 43 hemichannels. So what are these hemichannels? Connexin 43 is involved in the formation of gap junctions, and these junctions, they are critical, connecting, providing direct communication between cells. But these pathological hemichannels, which are not connected with the cell, they are connecting the cell with the cytoplasm. These are pathological pores. And this drug, called Tonabersat, it will act on these pathological hemichannels. And as I mentioned, it has been tested in different kind of eye disorders, and it was well proven that it acts through the NLRP3 inflammasome. What does it do? So basically, by blocking these connexin 43 hemichannels, we are blocking the release of ATP. And this will prevent the activation of the inflammasome. ATP is being released when the cells are being stressed, they are going through cell death, and obviously in a condition like MS and neurodegenerative disorders, there is a lot of release of ATP due to these processes. And ATP is being released from glial cells, but also from neurons. And it will activate the NLRP3 inflammasome. So we are trying to block the release of ATP, and then this will block the activation of the NLRP3 inflammasome and also release the cytokines and prevent cell death and pathological processes that are linked to inflammasome activation in MS. So in this study, after many in vitro studies, this was the first one when we used the drug in live animals, and also, as I said, to look at its effect in a neurological condition. And we used the EEE mouse model. To induce EEE, the animals are being injected with MOG and also with pertussis toxin, and basically this will induce a very strong inflammatory response, and then we administer the drug at two different time points. One, 3 days after the induction of the disease, and one, 8 days post-induction. And the reason was we were looking if the treatment starts later, then we can still have a positive effect, or this is not an option. So it was really important to test this. And also we tested 3 different doses of the drug, and the drug was delivered by IP injections. And this is a really high impact procedure in animals. And basically when we reach like stage 4, the animal has to be euthanized. We performed this work at the University of Melbourne. And basically my lab received the paraffin-embedded tissue, the brain tissue, and then we examined the pathological changes there, and then we also examined the behavioral effects of the drug. So we had all these treatment groups, but for the final histological analysis, we used the controls, the MOG-only injected mice, and the mice which received MOG treatment and this highest dose of the drug, because this was the most effective. And then we performed immunohistochemistry when we are labeling with fluorescent tags and antibodies, different kind of molecules. And I explain you what we have seen. So when we are looking the early dosing group, here you can see the MOG-treated animals. So these are the MS animals. And then they reach, the clinical scores are quickly increasing in these animals. And then you can see when they receive the drug treatment, these scores are much lower. As I said, the experiment is ending at this point because we reached that stage when we need to euthanize the animals. When we look at the late dosing group, you can still see the protective effect of Tonabersat. This is very important. So even when there is already a really severe disease pathology, the drug still works. And then when we compared the early dosing and late dosing group, there was not a big difference in clinical scores, but slightly the late dosing animals performed better, so we performed the immunohistochemistry experiments on these animals. So here I'm going to show the results looking the corpus callosum, and this is the largest white matter tract in the brain. So we see the most significant changes in the mouse brain in this region, but I should say that all the brain regions are affected. And we also examined the striatum and cortical regions, and you see significant inflammation there, but also what you see is the loss of myelin binding protein. So this is a normal control. You can see that the white matter and the myelin sheets are like nicely labeled, and then here's a loss in the MOG-treated animals. And then when we give Tonabersat to the animals, then they have close to normal MBP staining. And then we looked at the NLRP3 activation. And I should mention that when the inflammasome is not active, the labeling is not so obvious. You don't see much on this panel here. But when it becomes activated, then the complex comes together, so you see these bigger particles labeled, and that's what we see when we induce MS in the animals, and then you can see that these are not observed in the Tonabersat-treated animals. And then we also looked at the activation of glial cells. Here, this is a microglia staining, and in a normal control animal, you just see a few cells labeled. This is absolutely normal. And then in the MS animal, you can see like a very intense labeling, but as I mentioned, not only in the white matter, but all around in other regions as well, but it's like most significant here. And then in the Tonabersat-treated animal, we see a reduced labeling of IBA1. And this is just a quantification to show the results. And then here you can see the astrocytes. Again, like in a healthy control animals, you just see a few astrocytic processes labeled, and then very intense labeling in the MS animal. And then Tonabersat effectively reduces the activation of the astrocytes as well. And then this is again the quantification of the results. We also looked at the expression of the connexin 43 because this is our target molecule. We didn't find any significant changes in the connexin expression, but this doesn't mean that there is no difference in activation. And also with this staining, we can't differentiate between hemichannels or the gap junction labeling, those channels which will form gap junctions. But it's still important to check whether there is expressional change of the target in the disease model, because if you see that, for example, like most of the connexin 43 hemichannels are lost, then it's probably not a great therapeutic target. But in this case, we don't see expression changes in connexin 43. So based on these results, we see that Tonabersat is really a promising approach. I would like to mention that there are so many drug development drugs are under development targeting directly the inflammasome. They are not approved by the FDA at the moment. Tonabersat is very close to FDA approval. It's undergoing now phase 2b clinical trials. And we are hoping if it will be approved for those eye conditions like diabetic retinopathy or macular degeneration, then basically starting a clinical trial for MS, it will be a much easier process. So I would like to thank everyone who has been involved in this project, especially Professor Helen Danish-Meyer and Professor Colin Green, and also the students who were involved, Bavia, Emily, and Antonio, and the technical help from Jennifer and Christina. And thank you so much for listening. Yeah.